Hey, you want to be friends? Let's form a bilayer, and here's to section 2.3 das años, plasma membrane. Note disclaimer.
Let's talk about the plasma membrane. Please note when we're talking about the plasma membrane, we're talking about it in terms of animal cells. Note that plant cells have what's known as the cell wall. So when we're talking about the plasma membrane, we're only referring to it as the main barrier of the cell, and that's in reference to animal cells. What we mean by barrier, we mean that it's selectively permeable. Remember, the cell has certain organelles internally. You have the Golgi apparatus, you have the mitochondria, you have the endoplasmic reticulum. All of these organelles work together like a factory to ensure the safety of the cell itself. And the plasma membrane is a key part of that. The reason why is the plasma membrane basically ensures what enters and exits the cell is safe for that cell. And it separates the cell's internal environment from the external environment, regulating what goes in and what exits. The cell membrane also allows for internal, what we call homeostasis. And that means that it allows for pH balance and ion balance and all the things that it needs to do to ensure the safety of its cell. It allows nutrients in and removes waste, and it receives signals from other cells. In fact, there's a whole unit dedicated to this called cell signaling, in which cells communicate with one another. So the plasma membrane ensures this proper communication within not only the cell itself, but with other cells. Remember, cells make up tissues, which make up organs, which make up organ systems, which make up organisms, which means that all cells within an organism have to interact with one another. And they do this through cell-to-cell communication. Now let's talk about the plasma membrane itself. The plasma membrane has other, if you remember, we talked about macromolecules. Well, the cell basically has the plasma membrane, which has phospholipids, proteins, cholesterol, and carbohydrates. These all work together to basically ensure that the plasma membrane is working properly. Now let's talk about what's known as the phospholipid bilayer. We're going to talk about this more in just a second, but what happens is, if you remember what phospholipids are, they have a phosphate head and a fatty acid tail. And phospholipids are unique because their heads are hydrophilic while their tails are hydrophobic. So what we mean by that is the phosphate part is water loving, while the fatty acid part is water hating. And remember, the cell is filled with what's called the cytosol, which is a gooey fluid consistency. So you're not going to want that fatty acid side to face that part of the cell. Remember, our body is composed of water and we have cells inside of it. We wouldn't want water-fearing structures to be facing the water portion of us. So instead, what's formed is the phospholipid bilayer, in which the phosphate heads basically formulate on both the interior and the exterior side, the intracellular side and the extracellular side. And internally within that, like a sandwich, is the hydrophobic side. Now in there you have those phospholipids, those proteins, those cholesterol and carbohydrates, and they all work together to ensure what enters and exits the cell is nutrients that come in and waste that comes out. Remember, this all ties in not only to section 2.1, but 2.2, where we talked about cell size. Remember, cell size is dictated by what enters and what exits the cell as well. So this is important to remember, that the plasma membrane plays a really key role into that.
Unpredictable? Please, I am the balance.
Let's talk about the plasma membrane and the understanding of it being the gatekeeper of homeostasis. Homeostasis is a term of basically saying that you're trying to maintain a sense of balance within the cell. So if you had a picture of the cell, you have the inside of the cell and the outside of the cell, and both of these parts are water contained, so they have some sort of liquid.
Because of this, I want you to think of yourself looking at the cell and zooming in and seeing the plasma membrane from the outside. So you have the extracellular, and it formulates, meaning the phospholipids formulate a sandwich where the outside is like the bread and the inside is like your peanut butter and jelly. So on the outside, you have the phosphate heads, which are hydrophilic, which means water loving, and they're on both sides here. Internally, you have the fatty acid tails, so that's these right here, and those are hydrophobic. The phospholipids basically are organized in this way, where their heads are orientated out on both sides, both extracellular and intracellular, to be facing towards that water. And inside, it's meant to be hydrophobic, so it could be what we call selectively permeable, which basically means it only allows certain things to go in and out. And they're there to basically stabilize the internal environment of the cell. So if it is a small object, it can go inside usually, not always, but that's the selective permeability part. But however, if you have something that's very polar or large, it doesn't really favor passing through the bilayer with ease. Instead, what you have are various proteins. So, for example, you can have a protein that is all the way through, or you could have what's called a channel protein, or you could have something that's just sort of floating internally within the bilayer as a signal. And you'll talk about cell signaling in a later unit, but what you need to know is there's different types of proteins that help assist. For example, the channel protein will allow for ions with this channel gate. And so they allow the really hard material that can't go through to go through with assistance with these. The membrane is very flexible, and so moving lipids and proteins allows for the selective transport and the shaping and the communication and protects basically the inside of the cell. Sometimes you'll see a little hanging portion, which are basically carbohydrates. If you were to zoom in, you'd see the six-ring structure that is there. And that, remember, from your macromolecules is carbohydrates. And they're important because they play in cell recognition and communication. And they're found in what are called glycoproteins and lipids. So you'll see these structures where they're falling on the outside and just sort of attachment style, like a hook. And so we talked about phospholipids, carbohydrates, proteins, and let's talk about cholesterol. So cholesterol you might think is really bad because that's the stigma. However, when it comes to the phospholipid bilayer, you'll notice that they're there because you have to think of cholesterol as being fluid and stable when there are temperature fluctuations. So if something's really hot or really cold, you'll see cholesterol really being there to be that barrier of assistance to help in that flexibility. So that is the purpose of that structure. Now, if we were to talk about the fluid mosaic model, this is a model that we use to describe the phospholipid bilayer because it's the mixture of those proteins and lipids and carbs, and they form like a patchwork. So if we were to zoom out, you'd see something like this. And if we were to sandwich the phospholipid bilayer, you'd see that it sort of is not a perfect circle when it comes to the cell. Remember, the cell is shaped sometimes in odd shapes, and that's partially because of the phospholipid bilayer because it's fluid and somewhat movable. So it sort of fluctuates inside. Proteins can float within the membrane. So you could see there are different types of proteins that we talked about before. And that's because they're mosaic. They're flexible in nature with these proteins and they allow for various cell functions. If you look at this picture here, you see the phospholipid bilayer. And if you zoom in, you'll see that six-ring structure. Those are the attachments. And if you remember in the other photo, you had a protein that was attached to that, or sorry, lipid attached to a carbohydrate. So when it's called a lipid attached to a carbohydrate, we call that glycolipid. Then, if you have a protein that is embedded from end to end, we call that an integral membrane protein because it goes from one end all the way to the next. If it's a glycoprotein, that's the difference between a glycolipid and a glycoprotein. So a glycoprotein also has a carbohydrate attached to it. However, it's attached to a protein, and that's the difference. So a glycolipid, just to end any confusion. So if you think of the phospholipid with the heads, think of a carbohydrate attaching to that part. So that's why they have that picture here. So if you think the head, you think, okay, glycogen, glycolipid. Then if you think of, so that lipid with carbohydrate, whereas if you have that structure of a protein and it's attached to the same carbohydrate structure with the 6 rings, that's going to be called glycoprotein. You could have cholesterol, which sort of floats in between. And then, as we said, you could have the peripheral membrane protein, which sort of stays at the tail end. And then with all this, you have these weavings that are there. And it's important to note that the integral proteins sit within the phospholipid bilayer, and they're spanning across what we call as transmembrane proteins because they're on both, they go throughout the entire phospholipid bilayer. And sometimes the position of how they're involved depends on that R chain, because remember, amino group acids have different R groups. So that's important to note when you talk about that. If you're talking about the plasma membrane in general, this is a great table just to summarize what we went over. It talks about all the different parts of the fluid mosaic model. So you have the phospholipids that help form the bilayer, and they are separate versus inside and outside with the shape. We talked about the proteins and where you would find them, steroids and how they're inserted in phospholipids, glycoproteins, how those are glycoproteins, meaning carbohydrates with proteins, and then glycolipids, which are lipids attached to carbohydrate chains. We didn't talk about the functionality too much, the difference between glycoproteins and glycolipids, but glycoproteins are sort of for cell identification, communication signaling, and cell recognition, cell-to-cell interaction is the glycolipids. So those are the differences between the two. This is a great summary just to help you better understand the similarities and differences. Now, when we're talking about the different types of proteins, they could use other terms. For example, you could have something called an adhesion protein, and basically there are connections between the cells. You could have receptor proteins. So those are hooks on proteins, things like hormones that receive those different hormones. You could have transport proteins that basically are going to be embedded within, that are going to either be passive or active with ATP, and channeled proteins, which will be more specific, that allow those molecules or ions to move across. And we'll talk about that more in detail, more in Unit 4. But basically, these are just there so that way you can have structural fit that enables transport, signaling, anchoring, and enzymatic activity. Now, the thing is with proteins, remember, proteins have folds. And if we have something called misfoldings, what ends up happening is you have a failure of function, which is kind of not a great thing to happen because that could eventually lead to disease. So if we dive in more into the types of proteins, I wanted to point this out that you could have a channel protein, and if it has ATP, it's considered active. You could have it without ATP, and that would be considered passive transport. With the passive transport usually is without any sort of assistance, like a protein. So the thing is, you want to remember you're watching that selective permeability to allow for the substances to go across without that protein. So small nonpolar items will tend to freely move across the membrane, which is this first image here. But if you have polar or charged molecules or large molecules, they'll need some sort of transport protein. So you'll talk more in detail about passive versus active transport. And for example, you have water molecules that basically exist to help larger quantities of items pass through as well. So these are just different things to help you out. Now take a moment to read the following question, pause, and then let's answer this together. So the question states cholesterol is a component of the plasma membrane. What is the primary function? So take a moment to answer the question, and the correct answer is C. So the goal is to basically make sure that you're modulating proteins, modulating the membrane. So what do we mean by that? You're not increasing or decreasing the fluidity. It's a cross between the two. So A and B are not going to be the best answer. If you remember, cholesterol has to maintain the fluidity, basically not just by increasing or decreasing at all temperatures. It basically modulates the fluidity by interacting with the different phospholipids. So at high temperatures, cholesterol tends to restrain excessive movement of phospholipids, preventing the membrane from being too fluid. And at low temperatures, cholesterol prevents phospholipids from packing too tightly. And D wouldn't be a correct answer because it says that you're serving as primary energy source. And remember, ATP is energy. Now, this question basically hones in not only on Unit 2, but previous units as well. And that's why this question was here. You might be thinking, what do you mean? We talked about cholesterol, but we did not talk about high or low temperature. And you are correct. But if you remember when we talked about cholesterol, when we talked about the macromolecules that it's associated with, you want to think about the fact that cholesterol, when it's there, it has to interact differently with high and low temperatures. And that's just a type of lipid that is hydrophobic, right? So it's water-fearing. So if you remember in lipids, you have this idea that lipids can be dependent on this action of what's really happening if it's a high temperature or a low temperature. Is it a double, if you remember double, if you have the double lines or you have triple lines, how is that going to interact? And cholesterol is unique because it really allows and maintains fluidity based on the temperature that's involved with the phospholipid. So that's just thinking outside the box based on the information you learned on Unit 1 as well.
Appreciate you riding along for our cosmic journey. If today's mission brought you the knowledge that slaps, make sure to hit that like button to boost that signal. Subscribe if you're locked in for our next launch. And don't miss the link tree. It's got all the goods for AP Bio, PSAT Math, and SAT Math. Before I sign out, make sure you're all in one orbit and diving in. Killing out. Till next time.